So this is in the case of Chlamydomonas. In the plant, PSAI is chloroplast encoded, but some peripheral subunits are not completely conserved. So PSA A and B chloroplast encoded and homologous polypeptide. So very similar, but it's not exactly the same. So these two subunits are migrated in a similar position, so you cannot distinguish two subunits are co-migrated. And this is very hydrophobic protein, 11 hydrophobic helices, transmembrane helices. So that's why this protein is, if you separate on the gel, the diffuse, show the diffuse band. This is one of the characteristics of the very hydrophobic proteins. And also there are many relatively smaller subunits, sharp band, there's so many, and very small subunit. And two PSA I and J are very small, about 4 kilodalton. So it's usually difficult to separate on this gel. So we detected this by immunoblotting using antibody. In this preparation, PSA N and O are missing because these two subunits are very weakly bound to PS1. So doing a purification, these two subunits are lost. Okay, you need to be a very special use a special condition to isolate PS1 containing these two subunits. So because this region, many subunits has a similar size, so some polypeptide is very closely migrated or overlapping. So it's difficult to say how many, but actually by assigned by immunoblotting or mass spectrometry, there are 9 LSCA subunits. Usually higher plants, vascular plants contain only 4 LSC1. But Chlamydomonas may be adapted to the condition of lower light to grow. So, thank you. It's nice. So, it's the same as other green algae, contain more LSC1, light harvesting too. Harvest more light. Okay, and there are several small PS1 subunits. So these are the separation pattern. This is a little complicated. For example, LSCA 635 is migrated very closely, like a broadband, and LSCA 7 and 2 migrated at the same position, and LSCA 8 is a little bit lower, but it's overlapping. LHA 1 and 9 are overlapping, it's the same position, something like that, and L, H, E, G, C, K. So this is a, although this is used very high resolution gel, it's a long gel and very special content of buffer. So this is one of the very best separation for us, but still many subunits are separated very closely or overlapping. Okay, so this is a PS1 LH1 supercomplex. Yeah. Okay. So first, we want to. I want to show you that how this 9 LH1 complex are accommodated in this PS1 LH1 supercomplex in Chlamydomonas, because this part is very much similar from the cyanobacterial PS1 core and to vascular plant, algae also. So this is almost we know. Only some peripheral subunit are some missing or additional subunit. So this part we focus on this region. So first, we want to know this 9 LSC1 is present in PS1-LSC1 supercomplex stoichiometrically. Okay, you mean each subunit, one copy of each LSC1 present in this supercomplex, or not. Some are more than one copy, or some are half copies and heterogeneously associated or not. So to determine biochemically, we labeled proteins of chlamydomonas with carbon-14 radioactive isotope. Carbon-14 is not often used recently, but you can uniformly label all the protein by labeling a carbon-14. Then you can estimate the amount of each polypeptide quantitatively, because staining, this is staining. The staining is not proportional to the size of proteins, because some amino acid stain better than the other. So depending on the amino acid composition, the staining is not equal. So from staining, you cannot estimate the stoichiometry precisely. So we grow the cells in the presence of carbon-14 acetate. Acetate is a carbon source, so we use the carbon-14 acetate in the medium, and then grow the cells 2-3 days. Then whole proteins are uniformly labeled with carbon-14. Then we isolate PS1 and separate polypeptide, then detect radio activity by X-ray film, or some more quantitatively by some intensifying screen, and then the machine estimate the intensity of the signal. Okay. So this is a result after. This is not staining. This is a autoradiography. So intensity, the signal indicate the intensity of the radioisotope, radioactivity. So the intensity of this band represents the amount of protein. So we can estimate amount of proteins in this complex. But as I told you that there are many non-LSC1 and also PS1 subunit migrated this region. So some polypeptides are very close or overlapping. So it's difficult to estimate, right? So we use 3 different separation method system. This A is you can separate LHC4, 6, 3, 5, 7, 2, and 2. 2 is a little bit diffuse, and 7 and 8 and 2. So LHC1 and 9, PSAD and something like that. So you cannot estimate each of the LHC1 by this separation. Then we used a different separation. We changed a little bit the buffer system of the gel. So relative migration of polypeptide changes. Okay. So here, LHC4 is here, the 6, and here 3 plus 5. This is 7 plus 2. This is 8, 9, 1. But PSAD and F are in the same, something like that. This under this A condition, these two are separated, but here it migrated on at one single band. But this one is LHC 1 and 9 was single band, but under this condition, separated at two bands. Something like that. And we could mostly separate it, combining these two, but still not perfect because LHC 7 and 2 still difficult. So then we did two-dimensional electrophoresis. Okay, so first is this page and second is this page is under the different gel system. So LHC4 is here, 3, 5, 6, and 7, 2, 8, 2 and 8, 2 and 8. So combining these three gel systems, we could almost perfectly separate and estimate each subunit. Okay, then this is the result. LHC4, based on the PS1 subunit, LHC4 is one copy. It's a different system, but it's almost 0.9 to 1.0, so almost one copy. LHC6 also one copy. LHC3 one copy. LHC5 almost one copy. So there's some deviation. And second, this base 278 is separated, but the other system it's three migrated together, so three copies. But these each one copy. LHC1 is somehow overlapping with other summit. So with 9. So LHC9 is almost one copy, and LHC1 was two copies. So it's not everything is not one copy. And this is Pearson subunits are one copies. Okay, and the different condition it's two copies, but it's separated as a single. So PS1 subunit has one copy, and LHC1 subunit one copy, except for LHC1. This is two copies. So PS1, LHC1 super complex contain 9 different LHC1 polypeptide, but copy number is 10 because LHC1 is two copies. That's our conclusion.
So can you please explain this temperature differences? Like what is the role of this? Yeah, yes, sir.
Okay. We use a high molarity trace this page. This is give us a good separation condition that if you run the gel, low temperature, high temperature, separation changes we found. So they're using the same electrophoresis condition, but under the very warm condition, and in the cold room, then we could separate differently. For example, this band, LHC1 and 9 migrate as a single band, but in the low temperature, separated. But high temperature, PSA-D and F separated as two bands, but low temperature is separated as single band. So combination of this different condition, we could separate all these 9 and 10, 11 polypeptides that are migrating very close to each other. That's possible. So, okay. So this is the biochemistry. So
the low molecular weight, they are very close.
Low molecular weight is, you mean, there's a two. This is we call the low molecular weight, but this is very low. This is a medium. So this region, there are many polypeptides. So we wanted to separate nicely this region. but others are separated very nicely. We couldn't estimate these two, too small. Okay, so but we are focusing on LSG1, so then 10 LSG1 subunits, and how they are organized in complex. Okay, so what we did was chemical cross-linking experiment. So you can the cross-link between neighboring subunit by chemical cross-linker. Okay, so if the 10 kilodalton polypeptide is cross-linked to 20 kilodalton polypeptide, that product is 30 kilodalton. Okay, that's a system. So only neighboring polypeptide are cross-linked. That's so that you can estimate where these 10 LSG1 are present in PS1, LSG1 supercomplex. Okay, so you don't need to know very detailed, but this is a result. Sorry. We make antibody first against each of LSG1. The LSG1 is very similar proteins. No, they are 9, but they are similar. So if you make the antibody, generate antibody against the entire polypeptide, so then this antibody may cross-react other LSG1. So, but for chemical cross-linking, we need a very specific antibody. So we use the synthetic polypeptide as an antigen. Okay, that's the region, the polypeptide sequence, amino acid sequence region that is unique in each. So it took many years until we get 9 different antibody against each of LSG1, but finally we could. So, okay, this is a little bit complicated, so you don't need to see everything. But for example, it's if you look at which one, this one, we use the LHCA3 antibody and PSAK antibody. Okay, and minus. So this is an immunoblotting of using anti-LHCA3. And this is immunoblotting of using anti-PSAK. Okay? And minus is no chemical cross-linker. Okay? Without the chemical cross-linking. And plus is after chemical cross-linking. Okay? Then, if you look at the minus, oh, this is okay. LHCA3 is here. And PSAK is a little bit weak here. Okay. And after cross-linking, we could see a new band, upper band here. And here, okay? And the position, when we detect it with LHCA3 and PSAK, the position is the same. This means after cross-linking, LHCA3 and PSAK was cross-linked and separated this position. So that means LHCA3 and PSAK are neighboring. So we did as many as we can, okay? So many different combinations. Okay, other example is, for example, Because sometimes the antibody cross-react with non-specific cross-react. So you have to eliminate artifact. Okay, I think this is clear one. LHCA2, LHCA9, without chemical cross-linking, this is original. This is original, okay? And after cross-linking, LHCA2 have shifted this tree here, and another one here. And also LHCA9 is after cross-linking, there's additional new band here, and a little bit here. So this means LHCA2 and LHCA9 cross-linked. And if you see additional upper band, that may mean the third polypeptide may be cross-linked very inefficiently. Okay, so these results.
So, so this cross-linking we are doing separately for each protein, or
Yeah.
it is in the cell?
We did many blots. Okay.
Yeah, like we take out the protein, purify it, and then cross-link, or like how are in the cell?
Sorry, we use the thyroid membrane most cases. That is that prevent artificially cross-linking. Because if you isolate PS1 and then cross-linked, you may see a cross-linking between different complex. To avoid this, we use the thyroid membrane most cases. Okay, this is a cross-linking, very classical method, but in the typical biochemical method. So this is the result. Okay, summary. For example, LHCA1 cross-linked with LHCA9. So this is the whole list. PSAL cross-linked with PSAO, like that. Then we made a model. Okay. This is the PS1 viewing from the stroma side. Okay, stroma side. And then PSA-A and the PSA-B. Green part is PS1 core subunits. F, J, N, O. This. And white circle is LHC1. So as I show you, LHCA2 and LHCA9 cross-linked like this. So, but from the other cross-linking, we accommodated that. 2 and 9 is located in a PSAG L08 site here as a dimer. And also LHCA 1873 and PSAN is here. If you isolate PS1, PSAN is easily lost, but if you use a thyroid membrane, N is cross-linked this region, so be sure that N is around here. And this for LHC1 arrangement is very similar to vascular plant PS1. And 5, 6, 4, 1 is located the second layer of tetramer of LHC1. But this is dotted line is we could not definitely assign one of the two LHC1 here. But from the arrangement, we are sure that second LHC1 is located here. So tetramer, dimer. It's arrangement LHC1. This is our biochemical characterization. Okay, so I think data of the location of PSAN is very precisely we could assign here. That is still remain completely determined, but I think this is one of the most reliable results. Okay, so is that okay? So this is a little bit the classical, traditional way of assignment. Then we could did determination of structure by using a cryo-electron microscopy. So maybe you know the cryo-electron microscopy. It's this can at a low temperature, the frozen sample analyzed. So by analyzing, we could determine all the subunit and also cofactors, pigments in the complex with very high resolution. Okay, so the question is our result by traditional and classical chemical linking by chemistry. This is correct or not? Okay. That was a very thrilling moment when we started this. Our result was completely correct or not. I don't know. It's, I think, okay, just continue a little bit. So here we isolate PS1, LHC1 supercomplex biochemically and then analyze the particle. So I'm not specialist of cryo-electron. This is a collaboration with a specialist. So if you take a photograph of the particle, looks like this. So many small particles you can see. Okay? But this doesn't say anything about the structure, of course. But somehow the software pick up all the particle. This is particle. Can you see like a particle? And then I don't know how many, 100,000 or so many particles. And also, of course, in the particle on the this is on the grid, the frozen. So particle maybe not always like this. Some are like this. No, many different orientation. It's frozen. That is important. And then software pick up and then sort it to the similar size, similar direction, no orientation, and then average. No, so different particle like this, then overlap automatically. They continue hundreds of thousands particles. So these are different particle, how to say, different orientation and averaged. So this is flat and the side view or many different things. Already, if you look at here, there's some core and then 2 layers like this. But this is different angle, 2 layers and core, something like that. Then from this data, some software assemble this 2-dimensional the picture into 3-dimensional. This process I don't know. But this is the results. No, what's the difference from here to here? Okay. This is the view from the stroma side. Okay. This is the core and LHC1, 2 layers. You see? Something you can see here. No? 4 particles. 2 layers. Then after analysis, refinement. So maybe it's difficult to see something like this. This part here is PS1 core, okay? And this part, LSC1. And this is also LSC1. LSC1, LSC3, 7, 8, 1. LSC5, 6, 4, 1. LSC2, LSC9 is tetramer, 2 layers of tetramers, and 1 dimer. Dimer is between the PSAG and PSA-Li, this region. So our result was correct. Very fortunately. We ourselves could confirm the results later, okay? Okay, so cryo-electron microscopy reveals a more precise structure. And also, this rod structure is transmembrane alpha helix, okay? So I showed before the LSC2 had 3 transmembrane helices, right? So this LSC1 also 3 LSC, transmembrane helices and a small hydrophilic small alpha helix is also present. It's a very overall structure is very similar to PS LSC2. Okay. But LSC2 here it's a little bit different. This contains 4 transmembrane helices. So this is what's new finding. Result. And this is a polypeptide and also cofactor, chlorophyll A and B and carotenoid. So this is only cofactors, pigments, like this. Yeah. So, well, all the region pigments are distributed, but it's 2-dimensional, so no, it's stromal side and the luminal side. We cannot distinguish between them. But it looks like rather uniformly pigments are present. And so the green is the chlorophylls in PS1 core, and this yellow chlorophylls in LSC1, light harvesting complex. So this is the side view. This is the thyroglobulin membrane. And there are many transmembrane helices. That's why PS1, LSC1 present in the thyroglobulin membrane stably. Okay. Some units are stroma side exposed to stroma side, and some are a little bit exposed to luminal side. Okay. And LSC1 is inner LSC1 belt is tetramer, and the outer tetramer here, and also dimer, LSC2 and 9 is here. Okay, something like this. And many pigments are present. So, structure of PS1 LSC1 supercomplex. definition is like that. It's a PS1 reaction center, PSA A and B, and PS1 core means 2 reaction center plus peripheral PS1 subunit. The vascular plant PS1 LSC1 contain 4 LSC1. So PS1 core plus LSC1 tetramer. Okay, and chlamydomonas has 4 tetramer, 2 layer of tetramer. So total LSCA 8, and 1 dimer here. So total 10 LSC1. So our biochemical data also was correct. Okay. So I was very happy to to know that. Yeah.
To the previous slide.
Sure. Sorry.
To the previous slide.
This one?
Yes.
Could you hold the microphone?
Presence of number of LLCs.
I don't hear very well. So
Yeah. Is
Okay.
it audible now? Yeah. No, I wanted to know, sir, in case of the plants, when we see that light harvesting complexes are relatively lesser than that of the algal systems, like Chlamydomonas. So when we see, what do you think, like the membrane biology, like particularly when we compare the plant chloroplast membrane, lipid membranes, and then algal lipid membranes and their surface area, how it is encompassing these more number of LHCs.
more LS1.
Yeah, like when
how?
we see here in here.
Yeah, there's a contact site, the inner and the outer. So interaction to stabilize this 2 tetramer is, for example, either hydrophobic interaction or some hydrophilic interaction like hydrogen bond or ionic interaction. This order together of this many weak interaction finally stabilize this. Very stable.
So will it lead to the thylakoid width to be more in algal thylakoids compared to the plant thylakoids? Like, because there is a lot of space is there, surface area is there in the plasma membrane lipid bilayer. No, I mean to ask, like, if the thylakoid surface area, the size itself becomes a bit larger in algae compared to that of plant because of incorporation of more of proteins, LSCs.
Yeah, of course, it's important thing is this interacting contact sites. So, and also this how to say, inner and outer is the circle is the size is different. So,
Yes, sir.
but these are completely fit for depend on the organism. So small change required for stabilize this structure. But we don't know yet what's the main key point. But it's
Yes.
right.
Yes.
Yeah, it's okay. Okay. So I think it's the time. So I still continue the next lecture. The Raja will take over. Then the following lecture today, I continue. So I just this, and also the next my lecture deals with assembly of this complicated structure. Of course, we don't know everything, but in a part of the assembly process we analyze. So your comment or questions are welcome.
Since I have one doubt, so regarding this LHCs, there are 10 LHCs, right? So in PS1. So we all know about the LHCs in PS2, how they are going to affect it in terms of any stress. They are going to either do state transition from PS2 to 1. So
State transition.
yeah, So in that case, any remodeling of this PS1 LHCs will happen, or they still be the same?
LSC2 associate this region in Chlamydomonas
Yeah.
to LSC2 trimer associate.
Okay.
But depend on the organism, sometimes the three LSC2 trimer associate, but it depends on organisms that grow under the different light conditions. So if the organisms grow in under the very low light condition, in general, they have a more light harvesting complex. So because probably Chlamydomonas is growing relatively, how to say, low light condition in the soil of the some not exposed, just shade of the other plant. So probably this is laboratory strain, but it's originally Chlamydomonas must grow. Probably they grew under low light conditions. So that's why it contains more light harvesting complex. And also under some condition, LSA2 trimer is present here. And also, of course, and there are many LSC2 detached from PS1 and PS2. So these LSC2 trimers may transfer excitation energy to PS2 or sometimes PS1. But we don't know exactly all the LSC2 how they behave. We don't know yet completely. We know only excitation energy transfer from the LSC stably associated with PS1 or PS2. So this part is rather flexible depending on the organism. So that is a kind of adaptation to different light conditions. Any other question?
Sir, kind of is one more components with plots.
Even the block?
Yeah, initial slides.
So?
Back.
Back?
TV. Yeah,
This one? Yeah.
so like how did you identify all the super complexes in PS1 LS?
Okay.
One?
Uh,
These are from like
These are obvious because it's high molecular weight. Of course, we detected this polypeptide by antibody.
yeah, but everything like
Everything
extras-
we assigned by immunoblotting
okay
because
based on
we
molecular
have an antibody. And also, in the beginning, we also analyzed by mass spectrometry.
Okay.
Cut this band and analyze the mass after digestion with trypsin. And because we know genetic information, so it's easy to assign. But these too small are only detected by the immunoblotting because too small polypeptide is sometimes difficult to assign by mass spectrometry. Somehow.
So these characterization are based on the mass spectrum only. Then
No, this immunoblotting, and this I think it's immunoblotting mainly because using antibody staining and then using antibody, so this band is specifically reacted with antibody against PSA-K. And sometimes this region is difficult, so we use the mass spectrometry. So not all the mass spectrometry, because mass spectrometry is very sensitive. So you have a overlapping protein like this. So then mass spectrometry data show that there are two LSG1 polypeptide. So we cannot distinguish which is which. But in Western blotting, very delicate difference in the migration position, we can distinguish between two overlapping proteins. So sometimes immunoblotting is more powerful than mass spectrometry.
Sir, this structural composition is similar in control and stress.
Sorry, so I don't.
This structural composition is similar in control and stress, or whether there is any remodeling happening during stress?
No, I don't understand.
During control and stressful condition.
Sorry?
During control and stressful condition.
Let's
There is any remodeling happening. Remodeling of this. Remodeling.
if you cell grows under different environment condition.
Remodeling happening.
Okay. The question is, if you grow the cells under different condition, like low temperature or high temperature, low light or high light, I haven't carefully analyzed so far, but it is said that LHC1 are very stably associated with PS1 core. So the amount and also localization of LHC1 are not changed. Probably that's true. But peripheral trimeric LHC2 are more flexibly shuttled. So depending on the growth condition, it may lost and it's associated or detached. It may happen. So this part are very stable, I should say. Okay,
Sir, what about the PS2 core?
PS2.
PS2 have any remodeling or possible?
PS2 core part is stable, but LSC2 is depend on condition, but it's there are many reports of PS2, LSC2 super complex, and some reports show that one trimer of LSC2 are present, and some report under milder condition, yeah, there are 2 or 3, and so the difference is ascribed to the difference of the growth condition or purification condition. We don't know exactly, but it's a kind of a competition of the number of trimeric LHC2. So if you report 1, and then others report 2, and others also report this, there are 3. So, okay, 3 is more fancy than 2 or 1, that kind of things. But they are seeing, I think, PS2 with 1 trimeric LHC2 or 2 trimeric LHC2, 3 trimeric LHC2, but they show maximum number of trimeric LHC2 in the paper. There may be more LHC2 with single trimeric LHC2, but they don't care very much at the beginning. They want to show that 3 is the largest for the publication. This happens. So this difference may due to the growth condition or isolation condition. That the isolation condition means it's an artifact, right? Okay, so this is very still remain to be determined. If we find a better, milder detergent, then we may have more PS2 preparation with more LHC2. That may happen. So it depends on the development of new detergent or new isolation technique. So it's biochemists sometimes not physiologists. So and also structural biologists also like protein they just compete with resolution. We have a better resolution than the other, something like that. But they don't care about the function or physiology of the protein. So this happens. Okay. So I stop it. Okay, right. Thank you.